پذیر نشد که این کار براتون انجام بدم. بنابراین مجبورم فقط صفحه کامپیوترم رو براتون شیر کنم تا بتونید در واقع فایل ها رو ببینید. خب خاطرتون هست که ما هفته گذشته در کجا درس دادیم بهتون؟ Yes, I can explain the lesson with English language. Please let me know. Do you know what was the last teach file for you? PDF جلسه قبل رو خواستم تو سیستم آپلود کنم که بتونید دریافت کنید اما نمیشه. I can't uploading the PDF of the last season. Therefore, I should explain or teaching the lesson as live. Okay. خب PDF رو براتون میفرستم. حجم فایل PDF مقدار زیاد هست. خاطرتون از جلسه گذشته تا کجا درس دادم بهتون؟ آره؟ آخرین مبحث که بهتون گفتم چی بود؟ In this season to teach you discovery of DNA and the history of discovery of the DNA. Okay, they extract heat-killed cells in the last experiment by Griffith's mouse experiment showed that there is a genetic material genetics that help bacteria to transform from a shape to another shapes. Okay, from S shape to R shape or different happens for the bacteria in the bacterial suspension. In this season, I want to teach about the history of discovery of DNA. The extract of heat-killed bacterial cells contain protein, RNA, and DNA. همونطور که خاطرتون هست تو آخرین جلسه که با هم صحبت کردیم اومدیم در مورد آزمایش گریفیس با هم صحبت کردیم و گفتیم که گریفیس کاری که بچه‌ها گریفیس کاری که انجام داد این بود که مشخص کرد که یک ماده ژنتیکی وجود داره که این ماده ژنتیکی میتونه باکتری ها رو از شکلی به شکل دیگه تغییر بده و خصوصیات و صفات اونها رو تغییر بده. حالا ادامه همون مبحث رو میخوام خدمتتون بگم. میگه the extract of heat-killed bacterial cells contain protein, RNA and DNA. Which of these substances were essential for transfer? سوالی که هست این است که کدوم یک از این سه فرم، ببین وقتی که اون آقای گریفیس وقتی آزمایش انجام داد 3 فرم ماده رو به دست آورد، موادی مثل پروتئین RNA و DNA. مشخص که کدومی که از این سه حالت، یعنی کدومی که پروتئین RNA یا DNA عامل ایجاد تنوع ژنتیکیه. As I said in the last season, in Griffith's mouse experiment, he could establish that there is a genetical materials that help the bacteria to transform its shape from S shape to R shape. But what was the genetic materials? The genetic materials could be protein, RNA, and DNA. Which of them is correct? And which of them are genetic materials? Proteins, RNA, and DNAs. Which of these substances were essential for transformation? And there is 2 questions. One of this question is, which of these substances were essential for transformation, and how did they figure out what is for transformation? In 1944, Oswald Avery and 2 of his associates, MacLeod and McCarthy, reported the nature of transforming substance. Okay, reported the nature of the transforming substance. Avery and his colleagues did their work in vitro using colony morphology culture media rather than bacteria in mice as evidence of transformation. This study provides the first experimental evidence that the DNA was genetic material. DNA transformed R-type bacteria into S-type bacteria. Okay, therefore, in Oswald Avery and his colleagues' experiments, they could put that DNA is a genetic material, and the transformation of the bacteria from R shape to S shape is connected to the DNA as genetic material. As you see in the cell, you can find 3 important material: DNA, protein, and RNA. But which of them is responsible for genetic inheritance between individuals? DNA or RNA, and as I said, Oswald Avery and his colleagues in his experiments, they could prove that the DNA is a genetic material. But what was the Oswald Avery experiment? Also, remind that 3S or a smooth DNA bacteria DNA was the genetic material responsible for Griffith's result. He designed 2 experiments. Now the number 1 and number 2. In number 1 experiment, the mixture of DNA and RNA, he prepared a mixture of DNA and RNA, and then treat this suspension with RNAs, and then add DNA to our bacterial suspension. When he did this suspension with RNAs, all of the RNAs were damaged, and all of them were eliminated, and therefore only DNA was remained. Then he added DNA to RNA, and then he cultured the bacterial suspension on Petri plates. After this cultivation, he only S bacterial shapes are used on this plate. In the second experiment, he prepared a mixture of DNA and RNA, and then treated this mixture with DNAs. When this suspension treated with our DNAs, all of DNA was eliminated in this suspension, and therefore only RNA was remained. And then he added RNA to our bacteria, and then he cultivated the bacterial suspension on a Petri plates, but he couldn't see any S-transformant bacteria. Therefore, he concluded that only DNA is genetic material, and only DNA can transform the S-form bacteria to R-shaped bacteria or R-form bacteria. In this experiment, he could prove that the DNA is responsible as genetic material. Phage labeling. What was the phage? Okay, who can explain anything about the phage? Who can explain the phage characteristic? Can you hear my voice? Can you hear my voice? Okay. بعضی از دوستان مثل اینکه صدا ضعیف، صدای من ندارم. من پیام‌ها رو نگاه میکنم. برای همه خب چاره نداریم دیگه بچه‌ها، فعلا اینترنت سرعتش پایینه. کسی هست که بدون فاژ چیه؟ The phage. What is the phage? Phage. What is the phage? بچه میتونید بگید که فاژ چی هست؟ کسی فاژ رو میشناسه؟ Yes, the phage is viruses. The phage is viruses. Okay. And this virus can attack to bacteria, and the bacteria will be died when the phage attack to them. Okay. Valuable information about the nature of the genetic material has also come from viruses. Since phages consist only one nucleic acid surrounded by protein, as you see, it is a shape of the fungus. It is the shape of the phage, and as you see, the phage included proteins and genetic material as DNA. Therefore, you can see protein and DNA. Phages can insert its DNA to the bacterial cells, and he can replicate its genetic material and and its other proteins substance in the bacterial cell. Okay, I want to show you a video about the phages and everything about how it work.
The T4 phage initiates infection of an E. coli bacterium by recognizing cells.
It is a phage, and in this you can see the life cycle of the bacteriophage T4 viruses. Come to see with us.
Of its receptor of its long tail fibers. The recognition signal is sent through the long tail fibers to the base plate. This unravels the short tail fibers and irreversibly to the equal suffix. The base plate change.
As you see, the phage inject its DNA to the host, the bacterial host.
Changes confirm and the tail sheath contracts, causing GB5 at the end of the tail tube, not the outer membrane of the cell. The lysosome domain of GB5 is activated and degrades the
خب
periplasmic.
همونطور که می‌بینید بچه‌ها فاژ تشکیل شده از دو بخش، بخش پروتئین و همچنین آر‌ان‌ای و بخش دی ان ای و این قادر هستش که دی ان ای خودش رو به درون بافت میزبان تزریق کنه.
After the life cycle is complete, the host cell bursts and ejects the newly built viruses into the environment, destroying the host cell.
Okay. In flash file, I want to see you. I want to show you
DNA transformation involved. In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme that breaks down the host DNA into smaller fragments. DNA is replicated and protein induced. During formation of the mature phage, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. Bacterial DNA is introduced into the new host cell and can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
Okay. Do you have a question about the phage and its structure? Do you have a question about the phage and its and its structure? Okay. Valuable information about the nature of the genetic material has also came from viruses. Since phages consist only one nucleic acid surrounded by protein, they lend nicely to the determination of whether the protein or the nucleic acid is the genetic material. Hershey and Reese published in 1952 the result of research that supported the notion that the DNA is the genetic material and, in the process, helped to explain the nature of viral infection process. As you see in this experiment, they designed an experiment with 4 steps. In the first step, T2 bacteriophage is composed of DNA and proteins. As you see, T2 bacteriophage is composed of DNA and protein. To set up 2 replicates, label DNA with PCO2, label DNA with PCO2, and label protein with SCO5, and label protein with SCO5. Label DNA with SCO2, PCO2, and label protein with label DNA with P32, and label the protein with S35. In the three sections, infect by bacterial with 2 types of the label T2. 32 phosphor is discovered within the bacteria and progeny phages, whereas S35 is not found within the bacteria but released with the phage. Okay. Experiment that showed DNA to be genetic material of T2. In the first stage, he labeled the DNA of the phage by P32, radioactive phosphor, as 32. And in the second experiment, they produced a protein, and this protein has a sulfur radioactive as sulfur-35. When these phages attack to the bacterial cells, we found that only radioactivity recovered in host and passed on to the phage progeny. But in the second experiment, radioactivity recovered in phage host and not passed on to the progeny. Therefore, he can conclude that the DNA is genetic material and not proteins. Conclusion about these early experiments. Griffiths in 1928 and Avery in 1944. They found that DNA and not RNA is determining agent. Hershey and Chase in 1953 concluding that DNA and not protein genetic material. Frankel, Contrat, and Singer found that RNA and not protein is genetic material of some viruses, but no non-prokaryotes or eukaryotes use RNA as their genetic material. Do you have a question? اوکی. پس اگه بخوام به زبان فارسی برای شما بگم، ببینید بچه وقتی که آقای گریفیس اومد بررسی‌هاش انجام داد، به این نتیجه رسید که یک ماده وجود داره که این ماده میتونه در واقع کار انتقال مواد ژنتیکی رو انجام بده. اما سوالی که بود این بود که اون ماده‌ای که نقش انتقال ماده ژنتیکی رو به عهده داره اون ماده چیه؟ بررسی‌ها نشون داد که در واقع سه تا ترکیب اصلی وجود داره، یا پروتئین یا آر‌ان‌ای یا دی ان ای. سوال این بود که کدومی که از این سه تا تونستن ماده ژنتیکی رو منتقل کنن. برای اینکه مشخص کنن که کدومی که از این سه تا تونستن ماده ژنتیکی رو منتقل کنن، آزمایشات مختلفی طراحی شد. مثلا توی آزمایش قبلی همونطور که می‌بینید اومدن یک سری فاژ رو گرفتن، یک از این فاژها رو مثل این فاژ بالا توی یک آزمایش پروتئینش هم پروتئین معمولی خود فاژ بود، فقط دی ان ایش رو رادیواکتیو کردن. خب و در یک آزمایش دوم باز همون فاژ معمولی بود، فقط در اینجا دی ان ای رادیواکتیو نشد، اما پروتئینش رادیواکتیو بود. بعد اومدن این در واقع تزریق دادن و باکتری‌ها در معرض این فاژها قرار دادن. نتیجه چی بود؟ نتیجه بود که مطالعات نشون داد آنچه که در واقع ریکاور میشه بعد از اینکه فاژ میاد سلول باکتری رو هضم میکنه و ازش میاد بیرون، آنچه که در واقع ازش ریکاور میشه همون دی ان ای چی هست بچه‌ها؟ همون دی ان ای رادیواکتیو شده یا لیبل شده است. این دی ان ای رادیواکتیو و لیبل شده است که در داخل سلول باقی میمونه و ساختار سلول باکتری رو تخریب میکنه یا به برق گیر میده. اما پروتئین ها این خاصیت رو ندارن. درست شد؟ و توی این دو تا آزمایش اومدن با هم دیگه مقایسه کردن، دیدن که آنچه که باعث تغییر شکل باکتری ها میشه، آنچه که باعث میشه که باکتری ها عملکردشون رو تغییر بدن، مواد ژنتیکی و نه پوشش پروتئینی باکتری. و در نتیجه تصمیم گرفتن یا تایید کردن آنچه که در واقع باعث بروز تغییر شکل در باکتری ها میشه، آنچه که باعث انتقال مواد ژنتیکی میشه، در واقع اون ماده ژنتیکی است و نه پروتئین. اینجا کسی هست مباحث رو نفهمیده باشه؟ اما میخوایم ببینیم که در واقع نوکلئیک اسید چیه؟ چرا بهش میگن نوکلئیک اسید؟ Okay? We want to know what is the nucleic acid and why it's called as nucleic acid. In 1870, a German scientist named Friedrich Scher had isolated the chemical found in the nucleus. These were proteins and nucleic acid. Friedrich Scher. He found that only nucleic acid is genetic material. He found that only nucleic is genetic material. پس در سال 1870 یک در واقع متخصص فیزیک آلمانی به اسم آقای میشر اومد در واقع مطالعه کرد روی هسته داخل سلول و اینکه این هسته از پروتئین و اسیدهای به دست اومده. مطالعات قبلی نشون میداد یا مطالعاتی بعدها انجام شد که در واقع اون پروتئین ها نقشی در ایجاد تغییر تحول نداره یا تغییرات ژنتیکی نداره و اون چیزی که عامل اصلی ایجاد تغییر ژنتیکی هست چیه بچه‌ها؟ اسیدهای نوکلئیک هست. و ما امروز فهمیدیم که RNA و همچنین DNA نوکلئیک اسید هستند. اما اینکه چرا نوکلئیک اسید هستند به این خاطر که هست اینکه اولا از نظر شیمیایی حالت اسیدی دارن و ثانیاً چون در داخل هسته قرار گرفتن بهشون میگن نوکلئیک اسید. پس مواد ژنتیکی نوکلئیک اسید هستن و دلیل اینکه بهشون میگن نوکلئیک اسید هم این هست که در داخل هسته پیدا میشن و همچنین حالت اسیدی دارن. خب این time to explain everything about the chemistry of nucleic acid. خب ما میخوایم الان در مورد ساختار شیمی اسید نوکلئیک در شما ساختار شیمی اسید نوکلئیک چی هست و به چه صورتی هستش. خب بذارید اول توضیح فیلم برای شما بزنم و شما فیلم رو ببینید و در قالب فیلم شاید در واقع راحت‌تر بتونید مباحث رو درک کنید.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick. The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Argaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar phosphate backbone with nitrogen bases project the end of each helix. One complete 360-degree turn of the helix, 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand region bonded to the bases or helix strand. The bonds hold the two strands of the double helix together. Adenine is always paired with a thymine in the other strand and a guanine in one strand. Always paired cytosine in the other strand. Note that guanine-cytosine bases form 3 hydrogen bonds, while adenine-thymine bases form two hydrogen bonds. This makes guanine-cytosine base pairs more stable than thymine pairs. Nucleotide strands also allow the sequence in one to determine the sequence in complementary strands. Two ends of a strand are not identical. Note that on one end of each strand, the 3' hydroxyl of the deoxyribose sugar is not involved in the backbone, or we say it's free. While at the other end of the same strand, the 5' hydroxyl deoxyribose end is free, or may contain a phosphate is free, and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are in opposite directions, or we say that they are parallel. In three-dimensional space, the two helices are equally spaced along the axis of the molecule. Such major groove and a minor groove are creating symmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
Okay.
This molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also B-form DNA. Now, move to a more simplified representation of DNA to discuss the details. Unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many units called nucleotide. A nucleotide has three components: carbon sugar, a phosphate group, and more possible nitrases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, there is a phosphate between the 5' one sugar and the 3' carbon of the neighbor. The sugar is called deoxyribose because it is missing a group at the carbon, which is ribose. Because of this, nucleotides in DNA, deoxyribonucleic, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by faster bonds. The phosphate group of one nucleotide binds to prime oxygen of the neighboring nucleotide. Thus, see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the strand, 5' to 3'. Looking within the sugar, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix. Simplified the representation by flattening and the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine purines, which have double rings. The geometry of the AT, or TA, and GC, or C base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angle spaces attached to the backbone. Other base pairs monomers, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions when the aromatic rings of the bases stack next to each other and share electron probabilities. The polarity of the helical storms forms cheating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair information. The groove is the base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in a sequence-specific or non-sequence specific manner. Proteins to position themselves correctly in the genome to out their designated tasks. Is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.
Okay. Do you have a question about this?
Structure of DNA. The DNA molecule is a polymer consisting of several thousand pairs of nucleotide monomers. Each nucleotide consists of components. 1. Pentose sugar, deoxyribose. 2. Phosphate group. 3. Base. Nucleoside means deoxyribose plus nitrogenous base. Nucleotide means deoxyribose, nitrogenous base plus phosphate. 1. Pentose sugar. Deoxyribose sugar is a pentose sugar with carbon atoms. 2. Phosphate. It is attached to the adjacent pentose sugar has their fifth and third carbon atoms by phosphate bonds. The phosphate and sugar constitute the back nucleic acids. 3. Nitrogen base. There are two types of nitrogen bases. 1. Purine, adenine, and guanine, and 2. Pyrimidines, cytosine, and thymine. Nitrogenous base is linked to the pentose sugar through glycosidic linkage to form one side. When a phosphate is linked to 5' OH to a nucleoside through phosphate linkage, a corresponding nucleotide is formed. Two tides are linked through 3', 5. Phosphodiester linkage form dinucleotide. More nucleotides join to form a nucleotide chain. A polymer has at one end a free phosphate moiety at 5' end of ribose sugar. At the other end, the ribose sugar has 3' OH group. The backbone in a polynucleotide chain formed due to sugar and phosphates. The nitrogenous linked to sugar moiety form the backbone. Model of DNA. The widely accepted model of DNA is the
Do you have a question about these clips? And found everything about the DNA structure? No? No, you know that the DNA is a polynucleotide, okay? And this polynucleotide composed of different monomers. That the name of this monomers is nucleotides. And each nucleotide is composed of 3 units. These units are 5 carbon sugars, phosphate, and 4 alternate bases. Okay. In this time, I want to finish the lesson, and I will continue the lesson in the next season. In the next season, I want to teach you everything about the DNA and the chemistry and the structure of the DNA. Okay, please write the number of this slide as verse 6, and I will teach you the nucleotide structure in the next season. خب پس تا جلسه آینده ما جلسه آینده بچه‌ها ساختار دی ان ای رو بهتون درس میدیم و این سری فیلم ها رو به شما نشون دادم، چون اگه بخوام درس بدم وسط درس مجبوریم کلاس رو تعطیل کنیم چون کلاس شما داره تموم میشه. ان شاء الله باقی مباحث باشه برای هفته آینده. موفق و پیروز باشید و خدا نگهدار. فایل های رو هم که میخواید رو فیلم ها رو میخواید یا نه؟ چی رو میخواید؟ متوجه نشدم. من کتاب بعد بهتون معرفی کنم بچه‌ها که بتونید برید کتاب رو بخونید. بعد کتاب رو هم بهتون معرفی میکنم. تو گروه براتون میذارم برید ببینید. یه گروه واتساپی تشکیل بدید، من رو هم عضوش کنید که انشاءالله بتونیم خدمت شما درس بدیم. فایل رو براتون بذارم اونجا. I will see you in the next season.